Good evening, everybody, and I thank the organizers for including me. I think what Rima's remarks were very appropriate in the sense that there's an urgency. Some of you have been with me on a road that just seems to keep going on and on without an ending, and I think that maybe now, we'll see some changes. We see the White House getting involved, we see legislature getting more involved. I mean, I tell my students, science just goes so far, but after that, you need money and you need support from the government. So I'm hoping that that is something we will get. I'm going to get a glass of water, by the way. I really have not put together formal talks, so I will just tell you where I'm thinking. First, let's go to the bacterium, and then I'll go to people and maybe go back to the bacterium. But the point is, if you're a bacterium and you're confronted by antibiotics, what do? You don't just gasp, and you start making more bacteria, but these are resistant to the antibiotics. So the bacterium has only one thing to do in life, is live, survive. We have lots of complications. They're simple. They have, one chromosome. But, they have trouble to deal with on a daily basis, especially with us who are trying to destroy them. And quite honestly, our newest goal is not to destroy them, but to bring them in and, like the new discoveries with microbiomes, use the bacteria as our allies in shaping the world and keeping resistance in control. How do bacteria become resistant? They acquire resistance genes. I don't know how much further I'm going to go, but I will not talk down to you about this story, but it's a fascinating genetics theory. I was doing work in France when I heard a talk on infectious antibiotic resistance, just even the term infectious. And a Japanese co-worker who worked with Tsutomu Watanabe, who I had the pleasure to work with, one of the forefathers of this field, was explaining that, in fact, the genes for resistance to antibiotics were being transferred to other bacteria. Now, all right, Josh Lederberg found episomes and going back and forth, but nobody had seen what they were seeing, which is, 4 different antibiotics resistances being transferred, and that they were transferred between a salmonella and an E. coli. And then as more and more data came out, more resistances were being found transferable. So hence, infectious disease, infectious antibiotic resistance. And that's a concept that I think you have to keep in mind, because bacteria are always trying to get ahead of us. And it's not as if I'm giving them a brain, but at least they have residual energy to be able to survive. The genetics are fascinating, and I always thought of them as sort of science fiction. I mean, in the days before this kind of resistance was here, we would hear about bacteria moving 4 different resistances from one to another. Wow. And I remember the first gels that Stan Falco did showing that this was an episome, or a piece of DNA that's transferable. And I guess now would be a moment to try to let you all know, who are not geneticists, that the genetics are fascinating, and we have lots of names for them. We have plasmids, we have integrons, we have transposons, we have all these names, because there's a variety of genetic mechanisms by which bacteria transfer resistances to others in the environment. But the critical feature I want to stress is that their transfer of DNA is to different bacteria that are evolutionarily more diverse than a dog and a cat. So you might say, gee, they're cats and dogs. But they're transferring DNA, and we're getting pictures of what they're doing. So that's a unique feature, and that's what we began to see and began to face. So put that on the blackboard to say we got to deal with that at some point. The other area has to do with the global feature of resistance. In 1981, we had a meeting in the Dominican Republic in which we brought several hundred people. It was the first time that a meeting outside the United States was sponsored by the NIH. And that was because it was in the Dominican Republic. You can't get much closer to the U.S. I guess you could with Puerto Rico. But anyway, so the meeting was held there, and who was there? There were South Americans, Asians, all bringing their unusual resistance genes or resistant bacteria. And among those who were there also were sophisticated geneticists and microbiologists who were using these to discover new antibiotic resistances and all the fame and glory associated. What was curious was that the discoverers were not really part of the process of finding out what's going on. And that meeting, I think, changed things because it established relationships between individuals in different countries, but not in the relationship of, I found this unusual resistance, I'm going to give it to this French person or this American to work on what is the reason. Why do bacteria, how do bacteria resist? They destroy the antibiotic. They don't let it get inside the cell because they pump it out. They substitute it with a new target, which is not subject to the resistance. So there are very many different ways in which the bacteria escape the action of the antibiotic. And that's also important to realize because that gives them so much more abilities to escape, as I was saying. The other feature of resistance that I think has not been shared largely is the enormous spread that resistance can take. And in that regard, where do these resistances come from? I'm asked that all the time. And I say, go out in your backyard and just grab some of the soil and you'll find it. And in fact, we coined this term reservoirs of antibiotic resistance, which was your backyard or someone's sandbox. So there's a huge quantity of bacteria out there with resistance genes that we don't even know about. They're waiting for an antibiotic to come on board so they can be expressed. In fact, that's how it happens, isn't it? What I call the antibiotic resistance equation requires two basic features. One is the antibiotic. If there's no resistance genes around, the antibiotic does its job, it's not resisted, and it does fine. On the other hand, if the resistance gene gets into the mix, now what you have is a resistant bacterium, and eventually a resistant infection, and resistance then takes over. So it's an interesting phenomenon on how you can develop resistance slowly, but essentially, in my mind, you need the gene for resistance, or genes, and you need the antibiotic to set the stage. There's a lot of talk about what's going on in the animal world in terms of giving antibiotics. And I think the One Health idea is really fascinating and best displayed and nicely displayed in our field, that is antibiotic resistance. So that these bacteria have a link, there's a link between people and nature and veterinarians and the world. And that connection is often through people or through the bacteria themselves. There's a lot of discussion on what is the fate of the antibiotics that are being given to animals for animal health and more so for growth promotion. And it's an area that we at the Center for Adaptation, Genetics, and Drug Resistance have been looking at for years. An argument that I've been working with the FDA for years, nothing has happened. Europe has banned this practice. The United States continues. There's more activity coming aboard, more resentment, more pleas to change things, but Europe doesn't have it. We still do. I can only hope that that will change. But what is the fate of that antibiotic? Well, it's given out in nature. So as many of us have done looking for, let's say, something simple like tetracycline is given to chickens and given to cattle and so forth. And what happens with the antibiotic is it gets spread into the farm, the water comes in the rains, it takes the antibiotic down into the ground, into other waterways, and disperses widely. Does anyone follow it? Not really. Does anyone know about it? Not really. I mean, not in the way of understanding how this happens. But the phenomenon exists. The second part of that, in which I hear often, is, well, you're treating and giving these animals low doses, that's important, of tetracycline. But the resistances that you're claiming are coming from animals are to ampicillin and to aminoglycosides and other. So how can that come out of dosing animals with one antibiotic? Well, Naomi Datta gave us a hint about that many years ago. She looked at the antibiotic resistance profile of women taking antibiotic for urinary tract infection. Where did she look? Not in the urine, but in the feces. And what she found was, with time, instead of just one resistances, the women were accumulating different resistances, where and how, on these plasmids, these DNA, these genetic elements that move bacteria, move resistances back and forth. And that's an important concept, because we are confronted with the thought, what happens after the antibiotic sees the animal group. But I think that what the studies we have done on the farms and other farms have shown that the use of a single antibiotic for 7 to 10 days will lead to emergence of resistance to that antibiotic, but with 7, 8, 9, 10 days, you suddenly see other resistances emerge. Fascinating. Where do they come from? Well, no one really knows, but some investigators in Tours, France, had an idea. They saw that the chickens that they were feeding antibiotic-laced feed were doing fine, but eventually were excreting resistant bacteria, not only to tetracycline, but to other antibiotics. So they sat back and they looked at the chickens for a while, and this is a true story, and they saw that the chickens didn't stop eating. Now, when they went to look for the mystery resistances, they figured a gram of feed should be enough, and they took a gram of feed and they shook it up with the medium, they plated it, they didn't get the antibiotic-resistant bacterium. But when they looked at these chickens eating merrily the feed, they realized they didn't stop at 1 gram. They went on to 2 grams, 3 grams, 4. So the next time they did the experiment, they took 10 grams of feed, and they extracted the resistant bacteria. So it's in the design of the experiment how you can really find the answers, and that we need to think more broad. This was a very simple-minded experiment, but it opened up, in my mind, the ideas of what were needed in order to confront arguments about using in the area of antibiotic supplementation. Harvey mentioned earlier that we had established, where is the... Oh, here, I'm sorry, all right. Well, when I put this together, I guess I thought I would be short. So I will say thank you very much, and I hope I'll have some chances later.